Thank you, Faith, for that kind introduction. And I certainly share the sentiments of enjoying our time we spent with Faith and the Changer Hub and the people in it. Part of what makes the journey worthwhile is the people. The science is great. The people are important. So I'm from industry, and we're talking to immunologists primarily. So I have tweaked my talk somewhat towards immunology and chosen some controversial topics, which tends to be the story of my life. And want to be as bold as to say there may even be some lessons for vaccinology from industry. We're African focused, we're based in Lesotho and South Africa. And for those young people, much to Faith's point earlier, we'd like to encourage you to consider how to think differently from the global north about how we address these challenges. We don't have to replicate, we don't always have to tech transfer. There are things that can get done differently here, like we've seen with Patricia in Brazil, and hopefully that message comes across today. We have a virtuous circle. We do quite a few different things. We try and fill gaps where our partners in government have challenges. So we're in diagnostics, we're in early warning. We've built systems to track certificates, COVID certificates moving across the borders in our region. We've built diagnostics labs, reference labs for government. But primarily our interest is in working within that ecosystem to ultimately develop therapeutics and vaccines that can impact within that ecosystem, impact on the mortality and morbidity of animals, people, and the environment. So we do some science, primarily in collaboration, but just to position us, we're somewhere between academia and the market. We're in this chasm of death that Musa referred to in his talk, of innovation that sits on the shelves of professors in theses and never gets to market. In South Africa, we work with a group that sits with one of the biggest biotech patent portfolios on the continent. This is at UCT. Very few of them have seen the market, and we're trying to get them into the market. So amongst other things, we do some drug discovery. We're the first group to do single-cell B-cell sequencing in quail. So we built a library of B-cells after being exposed to some of our vaccines, which we're mining for various binders. We've also been involved in downstream bioprocess optimization. You'll start seeing a pattern. I put the word focus in inverted commas. We're not particularly focused. We fill gaps where the gaps exist, and there are lots of gaps, right? So from antigen or antibody discovery through to bioprocess engineering, there are very few innovations in bioprocess engineering happening in LMICs. We've got a patent pending on something which we're currently evaluating with the South African government's funding at pilot scale for reducing the cost of producing monoclonals, primarily made in plants, but also in other systems. We also work on a vector for in vivo expression of biologics, vaccines and therapeutic biologics. And this is a patented vector that we are working on together with the University of Cape Town and Imperial College. We also manufacture passive antibodies. You'll start seeing a strong theme about passive immunity in everything we do. And we use this for feed additives already, but we have a vision to see this starting to replace antibiotic use in animal feeds. Passive antibodies made in birds, chickens, and other galliforms. We've also built some vaccine candidates during the pandemic. I think everyone tried to build a candidate. We built a chimeric FC RBD fusion candidate, and most recently, and I think aligned with our involvement with Changer Hub, we've been trying to convert our existing GMP facilities, which are currently making cannabinoids. There's a $30 million facility in Lesotho, and we're trying to retrofit this for antibody and vaccine manufacture. So why is it important to think about things like inheritance of acquired immunity, these sort of controversial fringe, if you want, topics. We are trying to find commercial viability in an environment where sustainability of markets is a really big problem for business. There is no collective to buy from you. The market is fickle. It's here today, gone tomorrow. The cost of production at the kind of scales we're looking at are often not viable. So we have to find innovative ways to get over those barriers. So one of the things that we've had to look at is trying to get private sector and government to work together to give us off-take agreements. And that's what we've done in Lesotho in South Africa, or in the process of doing. It's a long journey. And that helps deal with one aspect. Then cost of production is a barrier because we typically, our healthcare systems can't afford the kind of costs that come out of the global north's factories. So what can we do about that? And that's where we've got a patent pending in the downstream process. So how do you purify something more cost effectively once you've made it? Often 70% of your cost sits in how you purify, not in how you make it, particularly in the case of monoclonal antibodies. And then really the topic of the immunological part of the talk today is about antigen sparing. So if you can imagine that in a really good facility you can probably make monoclonal antibodies or vaccine at about $100 a gram. But if you're making a subunit vaccine, that gives you about 10,000 doses. Sorry, there's a zero missing in that calculation. 10,000 doses. That's if you're doing a 100 microgram dose. If you could reduce that dose to 100 nanograms, you can produce 1 million doses from that same gram. So surely it's worthwhile thinking about how I can adopt an antigen-sparing approach towards how I deliver my vaccine. Because from industry's point of view, threefold reduction in my costs, suddenly the viability changes dramatically. Right? We try and address costs at multiple points in this equation, but really the antigen sparing component is the one I want to talk about today. So, the inheritance of acquired immunity. There's a Darwinian evolutionary question that remains, I think, largely unanswered, which is why do we not inherit the lessons that the parent has learned from exposure to disease? By and large, why do we not inherit this? And why do we not pass it on to our offspring? It just makes evolutionary sense that if we want our offspring to survive what we survived, we need to pass it on. And arguably, for pediatric vaccinology, this is the fundamental motivation. Right? We can't easily pass on what the parents have learned during their immunological journey to the offspring. So we need to introduce ways to create that immunological response that's protective. But in a world where the genome and our increased understanding of the transcriptome and the epigenome and the immunome and the microbiomes and as a whole the metagenome, and how no longer is the phenotype described by the genotype or by the genome, but it's a complex interplay, right, between all of these omic studies or these omes within our beings that contributes to what the phenotype looks like. And phenotypes as diverse as immune resilience or mental resilience are described in these non-genomic manners these days, like the epigenetics, for example, of inheritance of mental disorder. So the immunome is not seemingly transferred to offspring. The immunome being the coding DNA for our library, our repertoire, our archives of B cells, T cell receptors, antibodies. And part of the reason why it's not transmitted is it's enormous. Arguably 7-fold larger than the genome of the human. So if you would think about the genome of the human as 3 billion base pairs sitting in 46 chromosomes, the immunome, if you want to pack it into the same cell, would be about 10 to the 16 base pairs, 1 million times more, arguably. We haven't quite figured out how much yet because we haven't sequenced that deep in the entire organism. But clearly there's no way as you can pack that much DNA and pass it on generation to generation. There's just not enough space in a cell, and who would want to carry all of this genetic baggage one generation to the next? But there are archives that the mother has that she passes on to the child. And bear with me, because I think these archives and this method of transfer can lead to inheritance of acquired immunity. And the way I propose that this happens is with 3D images. Now, some of you might have heard the structural biologist talk yesterday. I didn't finish the talk, but structural biology is all around the 3D image that we can evaluate of molecules and how they interact with one another. And I'd like to hypothesize, that these maternal archives are passed on mother to child in a similar manner, as a 3D image. So, what about passive immunity? There's a transfer, right? But it's transitory from our understanding. In birds and reptiles, 30 to 60 days at most. In mammals, it's absorbed through the placenta, at least antibodies are, and up to 24 hours post-birth. And then it's delivered in milk for months, sometimes years. So perhaps there's a mechanism we should be looking at for how evolutionary pressures have driven the survival of the species in the offspring in terms of exposure to disease. And indeed, it's a very important component of our protection in early life. Not obviously just for humans, but for all mammals. And I'd like to talk about immune complexing briefly, because I think it has relevance to how passive transfer leads to acquired memory. So immune complexes are very close to our research group because they primarily, from a business point of view, allow for significant antigen sparing. So there have been studies done in animal models where antigen loads in vaccines have been reduced down to the nanogram scale, and hence that number of 100 nanograms I threw out there, some even lower than 100 nanograms. And that's very exciting from a business point of view, because suddenly viability looks threefold better, right? What's important though about immune complexing is that immune complexing primes the immune system, broadly speaking, to a tolerogenic or an immunogenic response. And it's presenting antigen coated in antibody in a way that tells the immune system how to respond. Now, in a context of, and there's a lot of detail there that you immunologists understand, this is not new to any of you, right? I'm just framing it perhaps slightly differently. The current dogma is that this transitory transfer of antibodies in the placenta, but in milk, antibodies and cellular transfer, memory cells, B and T cells that are transferred into the milk for months, sometimes years in breastfed youngsters, are clearly providing a protection for the newborn in terms of disabling bacteria that are potentially pathogenic. They are modulating the immune responses in the child. But where's the learning happening for the infant? Remember that breast milk is not just IgA. The full isotype mix is there right the way through to late stage feeding of antibodies, IgG, IgA, IgM, even IgE. And why would you have this full isotope mix of antibodies in the milk if all they were there to do is to protect from invasion, for example, of potential pathogens? So at this point, don't forget that there's also C cells, T cells, CD4 positive and CD8 positive cells, some of which are targeted some of which are at the same concentration as in your blood, others are in a lower concentration. And interestingly, blood-derived B cells switch in the breast to a secretory phenotype and are malt-associated. So they attach to the lining of the intestine and they become resident in the associated lymphoid tissue. So if you can bear with me, because I can't draw a picture of this, I'm going to try and paint a picture with my hands and with my words around how we envisage this learning happening in the gut primarily of the infant. So we all understand that maternal antibodies are present and they have a repertoire which is tolerogenic and immunogenic. And arguably the most important repertoire is the tolerogenic immune repertoire, because what it allows the child to learn is the difference between friend and foe. Because in binding with different immune complex motifs to these candidate commensal bacteria and viruses, the whole microbiome, and those very few threats that exist in a healthy environment, what you're training the infant's immune system to do is to respond with a tolerogenic response to the commensals. And by and large, a well-established broad immune tolerance to a most diverse of commensal populations is your primary defense as an offspring to any potential pathogens that it may well encounter. Now, this model of acquired learning only works when the conditions are correct. And it's a combination of conditions. Number one, the maternal passive transfer includes exposure to antigens present in the environment. The antigens are present in the environment when the passive transfer is happening. And in that model, largely in the duodenum of the child, but elsewhere as well, you have this learning possible where antibodies are binding to antigen, most likely commensals, and are priming the immune system for tolerogenic responses. When a pathogen is present, these antibodies are binding in bigger complexes, which are priming the immune system for an immunogenic response. And so fundamentally, if you picture it as this 3D image, because understand there's no genetic transfer from mother to child around the immunome. But the way the mother's antibodies and T cell receptors and B cell receptors recognize an antigen is essentially a 3D model which then the naive immune system in the child is slowly learning to recognize. Maybe not with the first exposure, but over 9 months of multiple exposure potentially, if breastfeeding is for 9 months, repeated exposure with the assistance of maternal antibodies and cellular memory cells to modulate and teach the immune system how to respond in a similar way to the mothers. Unfortunately, fathers, you have no role in this process. It's entirely maternal driven. But I do believe therein lies some lessons for vaccinology, if indeed this is happening, and I think we're all aware of some element of this that is happening. We've spoken about placental transfer of not just antibodies, but all sorts of immunomodulatory factors. In this context, what's interesting for us is that it's potentially possible to deliver per oral antigen in complex with antibody, ideally of maternal origin, and not just antibody, but ideally the full repertoire of the maternal B cells, T cells, and the antibody isotypes, together with very low volumes of, very low loads of antigen to deliver a very efficient priming of the immune system. And all of this per oral and mediated at the mucosal interfaces of the gut. And I would argue that the kind of antigen exposure that a baby is being exposed to is in the picogram level, not even in the nanogram level. So there's opportunities in biomimetically understanding this mechanism to result in far more antigen sparing than we've even imagined thus far. But perhaps more importantly than just looking at the threats is understanding that in an environment where the diversity of the microbiome is being shrunk because of C-sections and lack of microbiome transfer, antibiotic use in early life, and just in general modern lifestyles resulting in a diminished diversity in the microbiome. Just understanding the ability to be able to manipulate the microbiome of the immunomicrobiome, so the immunobiology of the microbiome in an infant, is powerful and doesn't have to involve recombinant tech. It can involve something as fundamental as utilizing healthy wet nurses. We haven't spoken about wet nurses in a long time, but you can induce wet nurse lactation in healthy individuals that you can screen with tools we have available, and you can deliver a lot of this value around teaching infant immune systems with the hand-holding that comes from passive transfer with surrogacy. Surrogacy in terms of milk, surrogacy in terms of probiotics, surrogacy in terms of how antigen is delivered. All of which leads to a more biomimetic approach, in our view, as to how we've evolved to process antigen with this prolonged hand-holding that happens maternally to infant. And we're less, and it's a relatively non-invasive route. You know, you're eating your vaccine together with your adjuvant, which is the antibodies, and the possibility to address a broad scope of immune priming and immune focusing without having to focus on just a single disease is possible with relatively low-tech solutions. Antigen doesn't arrive in the infant's stomach in inactivated form. It might be immobilized in maternal antibodies, but there's no adjuvant. It's not killed, it's not recombinant, it's often live, right, and able to cause disease. And yet that's how the baby learns how to respond from its mother. And in theory, in some shape or form, it's possible to do the same kind of learning transfer in adults. It doesn't have to be mother to child, it can be adult to adult. And another interesting component for us is that passive transfer from human to human child is fantastic, but we've discovered that passive transfer also works from other species. So antivenoms in Africa are largely made in horses and are very effective at preventing death and morbidity from envenomation in humans. In this model here, Pseudomonas aeruginosa is treated with antibodies made in chickens and passively transferred. So there are methods to achieve some level of immune learning which are also possible passively through other species, which sometimes is a little bit easier than working with finding volunteers, for example, for surrogate breastfeeding. So I would suggest that some of the way forward are understanding from our omics technologies we have available the kind of maternal immunome that we would like to see transferred, and to build surrogate solutions like I've mentioned for some of those. In our lab, we vaccinate birds with candidate vaccines and we produce passive therapies which can be passed on. So all of these indicate that other outside the box thinking is possible on the continent, with or without patent protection, which allows for immunological interventions which can have huge public health impact without having to build a $300 million factory to make a vaccine. The immunology doesn't have to be less relevant, but we do need to think differently how to deliver solutions that are more impactful and accessible. So in conclusion, thanks to our key partners, including the Chandra Hub and the CSR in South Africa, and a couple of entities, the University in Lesotho, who work with us on many of these projects. Thank you.